Okay, hello everybody. My name's Rick Bushman. I'm co-chairing this session with Satya Dandekar. This is our last session, Vaccines and Therapeutics. It gives me great pleasure to introduce Roger Paredes as our first speaker in this session. We'll then have Bali, Pulendran, Alessandra Borgognone, and Shamsudheen Moidunny as our following speakers. So Roger is an MD PhD and HIV physician at the Hospital Germain Trias Puyol and head of the Microbial Genomics Group at the IrsiCaixa AIDS Research Institute at Badalona, Catalonia in Spain. Roger obtained MD and PhD degrees in medicine and surgery from the Autonomous University of Barcelona. He specialized in HIV resistance at the Brigham and Women's Hospital in Boston and Harvard Medical School. His team has demonstrated the clinical utility of HIV-1 deep sequencing in both high and low income countries. Dr. Paredes is a member of the WHO HIV Resistance Strategy Steering Committee and the International Antiviral Society of the USA, which annually produces the ISA USA list of HIV-1 drug-resistant mutations, which is used worldwide. He is co-author of the REGA algorithm for HIV resistance interpretation and virologist for the EuroCIDA European cohort. His group is presently leading pioneering research on the role of the gut microbiome in the pathogenesis of HIV infection and chronic inflammation. Earlier in the meeting, we've already heard about the really important finding in the last couple of years. They were the first to describe a link between sexual practices and the fecal microbiome composition. Dr. Paredes combines his research work at IrsiCaixa with a senior medical appointment at the HIV unit of the German's Trias Pujol University Hospital in Badalona, Catalonia in Spain. And so it gives me great pleasure to introduce Roger. His title is The Microbiome in HIV Vaccines.
Well, hi everyone. Thanks a lot to the organizers for inviting me to give you this talk. Let me start with this picture of the Milky Way, the Via Láctea, by saying that we have as many bacteria as stars in the Milky Way. So when all these bacteria interact among themselves, and also they do with the immune system. Therefore, it is not surprising that interactions are going to be very complex, but at the same time very, very interesting. Well, let's start by saying, there's some cumulative data suggesting that we could use bugs to modulate the immune system. Why is that? So it is well established that the microbiota modulates the immune system. So we know that different bacteria selectively modulate different immune pathways, including innate or acquired immunity, and among acquired immunity that includes cellular, humoral responses, and some bacteria exert pro-inflammatory actions, whereas others are anti-inflammatory. For example, you have an example here of segmented filamentous bacteria that stimulate Th17 cells, for example, or you have Clostridia, which also stimulate this Th17 or cells, and for example, Bacteroides fragilis, which is able to stimulate TGF-beta producing cells, also Th17, etc. So the microbiota, different bacteria selectively modulate different immune pathways, as we were saying before. The idea here is that there will be a number of environmental factors, including antibiotic use, diet, in the case of HIV, also sexual practices, which in a background of certain genetic susceptibility will lead to microbiome disturbance, evidenced by reductions in microbial richness, and also reductions in certain species or genus like Rominococcaceae, Lactobacilli, or increases in protobacteria and changes in the metabolites. All this leads to immune dysregulation, and it winds up creating disease. So also there are important interactions between the microbiome and the immune system in extraintestinal organs. For example, the liver. So LPS produced by the microbiota leads to TLR4 signaling, MAMPs produced by also the microbiota stimulate Kupffer cells. Probiotic have been shown to stimulate NKT cells, etc. But also another place where the microbiota might interact with the immune system is the central nervous system, essentially through short-chain fatty acids in what we call the gut-brain axis, which is extraordinarily complex, but also in the lungs. The lungs would be another area where either through short-chain fatty acids or through the presence of certain metabolites, different immune responses could be exerted in what's so-called the gut-lung axis. So also in the recent years, there is increasing interest in targeting certain immune functions by bacteria. So it is well established in this review from Skelly Nature Reviews Microbiology that certain members of the microbiota can stimulate the innate immunity through different systems and lead either to inflammatory responses, for example, or anti-inflammatory responses, depending. The latter would be exerted by Bacteroides fragilis, for example. Also, the microbiota has been involved in responses to immune checkpoint inhibitors. For example, there is a very interesting 11-strain consortium that is able to stimulate interferon gamma producing CD8 T cells through which accumulate inside tumors, and this leads to an enhanced anti-tumor activity, but also has been shown to have enhanced anti-infectious activity. Similar findings from Akkermansia mucinifila, and also reverse effects from, for example, Faecalibacterium. And finally, so microbes can stimulate T cell responses, but at the same time, and which is still also very interesting, is that microbes can also stimulate antibody-mediated responses and also plasma B cells. So such microbes are both controlled by antibodies generated by the body, but at the same time stimulate antibody responses. So in HIV, sometimes we want to induce immune activation, but sometimes that we want to do the opposite. So, for example, if we want to inducing immune activation might be good to eliminate cancer cells, might be good to improve preventive vaccine responses, or to attempt to cure HIV by purging the reservoirs. However, chronic immune activation has been shown to be detrimental because it accelerates aging, produces immune senescence, can also lead to hypersensitivity reactions or autoimmune disorders. So finding the right balance between the microbes and the immune system is absolutely key. What do we know in HIV? So the microbiome in HIV, this is a seminal paper that I always show. We have a figure from a seminal paper by Jason Branchley. This is what happens in the distal ileum of an HIV-infected patient after a few days following HIV infection. So huge changes in the gut-associated lymphoid tissue. And in order not to quote our own work, there is this recent excellent paper by a team at the NIH by Ivan Vujovic-Sijin and also Irene Sereti, showing two important things. One is that we had previously described that there was a microbiome composition associated with MSM sexual practice, and they find that as well, but they are also able, by using a very well-matched cohort study, they are able to identify specific genes that are enriched in people living with HIV, and also genuses that are depleted in people living with HIV regardless of sexual practice. And such genuses include often Ruminococcacea and also Clostridiales and Botulinum crossotus, but also Prevotella. So all these bugs are usually anti-inflammatory, and they are actually reduced significantly in HIV-infected patients. On the right-hand side of this slide, what they show is that When they look at these species, they can create an index associated with of microbiota associated with people living with HIV, and such microbiota is inversely correlated with CD4 T cell count nadir, or, for example, it is directly correlated with the number of comorbidities, so suggesting that such changes, which are HIV specific in the microbiome, are also are linked to problems, to clinically relevant problems. We observed that using shotgun metagenomics in the past, and were able to show a very tight relationship between gene richness and CD4 T cell counts, for example, and that seemed to be associated, or this is the hypothesis that we work with, that is associated with adaptation to oxidative stress following HIV infection. So, in summary, we have a microbiome that is able to induce or modulate the immune system, and we have significant changes in HIV infection that are also able to, which are might be associated with immune pathogenesis and clinical problems. So, okay, can we then, we want to explore whether such changes can also have an effect on different aspects of human immunity. This is a first proof of concept study I'm going to show you that we developed with the group by Bézin Combidier and in France, and also Christian Brandner in the IrsiCaixa, and Jorge Lama in Peru, in which this was the CATIVAC-03 study. Here we tested the CATIVAC-03 tested two routes of administration of an HIV vaccine, which is essentially an MVA plus an HIV insert, and they used transcutaneous but also the intramuscular route for administration. So what we did here, we evaluated the microbiome composition in both the skin and stool in these patients. So you can see how, first of all, the microbiome was was clearly different from skin and stool as expected, but also there were so the skin and stool genus correlated with skin and genus stool genus diversity correlated with expression of different blood genes. For example, there was the genus diversity of the Shannon or inverse Simpson had correlations with BLK, which is EFBF1, and IGLV861. All these are factors, these are genes associated with B cell responses. Interestingly, very interestingly, the abundance of Prevotella in the skin, but also the abundance of Eubacterium in the stool correlated very significantly with the presence of neutralizing antibodies against the MVA. So it is pretty apparent that the microbial abundance before vaccination was correlated with neutralizing antibody responses. When we combined blood gene expression with pre-vaccine host microbiota, all these factors we were able to identify a number of, a group of markers that actually predicted response to the MVA product that we were administering. For example, you have here a cluster of in ping of genes that were increased, for example, in protein transmembrane transport, translation, and transcription regulation, etc., etc. So another aspect which is also very interesting and that we are exploring, and now Alessandra Borgognone will show you with more details afterwards, is the idea that we can stratify T-cell HIV vaccine responses based on the baseline microbiome. Was a kick and kill vaccination approach in patients that had been previously included in the VSEN-01 clinical study. So these subjects were diagnosed very early and put on antiretroviral therapy during the first 6 months. During the VCN-01, they receive a dose of chat adenovirus and an MVA. They did not interrupt therapy at all, and they remain during more than 3 years with undetectable viral loads below 200 and CD4 counts above 500, always under INSTI regimens. Then, when these patients got into the VCN-02, they received consecutively first dose of an MVA-based vaccine, 3 doses of romidepsin, and another dose on MVA, and then went into monitored treatment interruption or monitored antiretroviral pause, in which they were followed until their viral loads reached 2,000. And then they were reinitiated on therapy. So here you can see the baseline characteristics of these subjects. All subjects were 13, and we had 3 individuals who were able to achieve spontaneous control of virus replication below 2,000, whereas there were 10 additional subjects in which the viral load rebounded very, very quickly. Alessandra will show you with more details of this study, but in a sense we found a highly significant difference in the ratio between Bacteroideales and Clostridiales, so individuals that were able to control virus replication even at baseline had much higher levels of Bacteroideales and much lower levels than of Clostridiales, and also Erysipedotricales than non-controllers. Suggesting, so these data has to be, Alessandra will show you more details, but this data has to be validated in the independent clinical trials, but it would suggest that it is able to stratify responses to vaccines, to the HIV vaccine, before or even identify patients who are more likely to respond to an HIV vaccine than others. Okay, so the microbiome has some impact on the humoral responses. The microbiome correlates also with cellular responses or immune responses to a T-cell vaccine. So the third question is, can we modulate the T-cell vaccine immunogenicity with bugs? In order to do that, we have started an experiment which is being run by Alex Orbera and Aleix Elizalde, which is still unpublished. And the idea here would be that we are testing Black 6 mice. We know we have previously tested the IrsiCaixa vaccine by Christian Brander in these mice, and we know it is immunogenic. So we have designed a set of experiments. The first one is we give them PBS. Then the second one, we just give them DNA, chimpadeno, and MVA vaccination. We expect with the HTI T-cell immunogen, we expect mice to develop a certain immune response, which we already know. And the idea here was to then do the same, but following continuous antibiotic treatment. We have to say that before, in order to homogenize the microbiome, all animals get through 5 days of antibiotics, and then they receive, they go through an FMT from a donor mouse. So they have the same microbiome to start with. We have data from these first 3 experiments, and based on these data, we will continue then by trying to apply different probiotics. Some probiotics like the B800 have been shown to induce CD8 responses. Others are able to induce, like V411, are able to induce Treg responses, so they should have a negative effect. And finally, we have V804, which is actually neutral and should not have any positive or negative effect on vaccines. So first of all, here you see differences. I want to apologize for the color code. This was not expected. I'm really sorry. So next time we will change it. So here you see the different differences in threshold and breadth and magnitude among female mice and male mice. So females usually have a higher response, but at the same time, if you look at the magnitude of responses, which is the sum of splenocytes that produce interferon gamma, you can see how female mice have higher responses if they receive antibiotics than those without antibiotics. Here, comparing differences between male and female. So these are the different epitopes that are being tested, and you can see how for taking all or both genera together, there are significant differences. So an increase in magnitude in different epitopes against GAG and POL, for example, but also BEEV here when we take into account both genera. And finally, you can see that most changes are actually coming from female mice. So what we're now doing is we're repeating these experiments. We are increasing the sample size and trying to see if whatever we find also in females can also be replicated in males. But the idea is that by modifying the microbiome, you can certainly change the response to HIV vaccines. Let me finish by telling you about a EU-funded project we were lucky to be awarded, and in which we have this is it's called Mistral, microbiome-based stratification of individuals at risk of HIV acquisition, chronic clinical complications, antimicrobial drug resistance, and non-responsiveness to therapeutic HIV vaccination. This is a large consortium, multinational consortium that includes different sites in Europe and the US, but also in Canada. So you have here the main the PIs. Now we have, for example, Rafik Sekali, who was in Case Western and now is moving to Emory, and we also have Adam Bergener, who is involved. Has a close scientific relationship with Christina Brolinden in Sweden. So the goals of this study are to identify novel signatures in any dimension of the host microbiome interaction associated with protection, response to therapeutic vaccination, development of clinical complications, systemic immune reconstitution, inflammation, and CD4 recovery following exposure to symbiotic products, provide a strong scientific rationale to design microbiome-based interventions, aim at preventing all these factors, to try to use these huge data set, sequence data set, to explore the microbial resistance, and finally to develop a practical cloud-based software tool for clinicians and researchers which will enable and facilitate microbiome-based patient stratification. These are the different work packages, which include mouse models, human cohorts, and randomized clinical trials. It will generate tons of data, and a main output that we expect to obtain is to obtain a list of microbiota-derived biomarkers of, as I said, acquisition, progression, vaccine responses, immune reconstitution, clinical complications. All these biomarkers will be published in an open access research data repository so that everyone can use it and can find new associations. And also we hope that all these data will lead to microbiota-derived treatments and interventions, and all that information will be used to develop and validate this subject stratification software called Thera, and hopefully this will lead to new medical models. Let me finish by saying then, as the summary, that the human microbiome can be used to stratify subjects according to their predicted response to HIV immunotherapy. This is what we think, and although we need to validate our findings and expand our findings, we think that this will allow identifying subjects who might not respond to immunotherapy. At the same time, it will allow the design for interventions in the gut microbiome to modify HIV vaccine immunogenicity. And of course, much more work is needed to identify specific molecules and mechanisms. We think that a view into a clinical future For example, we might be seeing the way that an individual who is a candidate to receive a therapeutic HIV vaccine, we might want to to perform a fecal microbiome assessment to ask ourselves, is there a favorable immune response profile? If it is, then we would just give him the vaccine. And if it's not, then we might be able to modify, or we might want to change that microbiome through microbial supplementation and ask again. Similar thing with chronic complications. We have patients and we don't know exactly if they will, with low native CD4 T cell counts, for example, but some of them will develop problems, others will not. So the gut microbiome might serve to prevent, to identify such additional risk and prevent changes. One last thought, I think that we are now like, the situation with the microbes is that we are now starting to see that some microbes develop certain effects which might potentially be beneficial. But this is like if we want to cure an atrial fibrillation with digitalis. It's like digitalis, the plant digitalis can help us slow the heart rate. But if we eat digitalis, that might be difficult to, it might be difficult to find the right dose and to predict what will be the dose, and we might reach toxicity also as well. So what we really need to do is we need to isolate the compound and then be able to produce this compound on its own, and possibly once we identify the specific molecules that are able to stimulate the immune system in the sense that we are interested, that's when the true microbiome vaccine medicine will develop. Let me end by thanking everyone. We collaborate with a lot of people, particularly the team in IrsiCaixa, Mark and others, Alessandra, Maria, and all of them, because I've been deeply related with COVID during the last times, and they held everything up. Also, Christian and all the founders. Thank you very much.
Hi, so it is my great pleasure to introduce our next speaker, Bali Pulendran. He is endowed professor at Stanford University School of Medicine, and also member of two departments, the Department of Pathology and Department of Microbiology and Immunology at Stanford. Prior to moving to Stanford, he was at Emory University, where he was professor of pathology and also director of the NIH Center for Systems Vaccinology, which he continues in the status of adjunct professor. Bali received his PhD in immunology from Walter and Eliza Hall Institute in Melbourne, Australia, and postdoctoral training at Immunex. His research is focused on understanding the mechanisms by which innate immune system regulates adaptive immunity. And he is identifying mechanisms to design novel vaccines. Bali has been actually pioneer in developing and integrating systems biology approach to predict the efficacy of vaccines and also identify new molecular correlates of protection. He's very accomplished scientist. His work has been published in Nature Science, Nature Immunology, Nature Medicine, journals like that. He's recipient of numerous grants, numerous awards, serves on several editorial boards, and has many grants, including NIH Merit Awards. Bali has also served and continues to serve on several advisory boards, including Keystone Symposia. And lastly, Bali is one of the highest or most cited researchers, top 1% of researchers in their subject during the past decade. So it's a great pleasure to welcome Bali for his next presentation as invited speaker in this session.
Good afternoon. I'd like to thank the organizers for inviting me to participate in this symposium, and sorry that we cannot be together because of the coronavirus crisis. Today I'd like to talk to you about some work we're doing in my lab to use systems biological approaches to understand the mechanisms of vaccine efficacy. Vaccines have been described as one of the most cost-effective public health tools in history and have saved countless lives from the specter of infectious diseases. Yet, to immunologists, vaccines represent incredibly powerful probes on the human immune system. Why do I say that? I say that for several reasons. First, vaccines can allow you to perturb the human immune system in a very synchronized way. Infections and autoimmunities and cancers and allergies can also perturb the human immune system, but one can never know with any degree of certainty precisely when the perturbation happened. With vaccines, however, you know exactly when the vaccine went into the body. Secondly, the advancement of high-throughput technologies such as transcriptomics, metabolomics, epigenomics have really allowed one to analyze the behavior of cells, genes, metabolites at an unprecedented degree of precision, so that you can really follow the immune response to vaccination with time at multiple levels of the biological hierarchy. Thirdly, is a practical reason. We all get vaccines. Vaccines are administered widely to very diverse populations, ranging from the very young to the very old, to those of us who are susceptible to autoimmunities or allergies, cancer patients. So the incredible diversity of phenotypes, human phenotypes, that is manifest in such diverse populations can be understood by perturbing the immune system with vaccines. Fourthly, vaccines represent a diverse array of viral and microbial stimuli, ranging from live viruses such as smallpox or yellow fever to inactivated vaccines, RNA vaccines, DNA vaccines, recombinant vaccines given with adjuvants, without adjuvants, carbohydrate vaccines. There's a whole array of different stimuli that are used to perturb the immune system. So this gives us a wonderful conceptual framework with which to probe human immunity and to understand how the host genetics and the environmental factors, as well as the microbiome, impinge on the immune physiology of human beings. Several years ago, we performed a study in humans to use systems biological approaches to analyze the innate and adaptive responses to vaccination with the live attenuated yellow fever vaccine. The yellow fever vaccine, or YF17D, is one of the most successful vaccines ever developed, having been administered to over 600 million people globally and having an efficacy of greater than 99.9%. Yet, despite its success, very little is understood about the mechanisms by which it stimulates protective immunity. So we performed a study, a phase 1 study, in which we vaccinated people with YF17D and analyzed the innate and the adaptive responses, as shown in that diagram. In Rafi Ahmed's lab, they analyzed the magnitude of the tetramer-positive CD8 T cells, as well as the neutralizing antibody responses. In our lab, we analyzed the early innate response that occurred within the first few days. And in particular, we looked at the changes in the gene expression profiles induced by vaccination, as well as the changes in the dynamics of innate cell populations by flow cytometry, and analyzed the changes in cytokines and chemokines by Luminex analysis. We were able to define an early signature, a molecular signature of transcriptional changes, if you will, that correlated with the ensuing CD8 T cell response and the antibody response. We were delighted to observe that a similar and independent study done by Rafiq Sekali and colleagues came up with pretty much identical signatures to what we had seen before. Furthermore, we used machine learning techniques to define a focused gene list of predictive rules, if you will, that could be used to predict in an independent way the magnitude of the CD8 T cell and the neutralizing antibody response in a completely independent trial that was done some 1 year after the completion of the first trial. So this study represented the first example, the proof of concept of the utility of applying systems-based approaches to defining predictive signatures of immune responses to vaccines. And in work that I'm not going to discuss today, This study has also revealed many interesting mechanistic insights about the mechanisms by which this vaccine is stimulating such effective immunity. Subsequently, we and a number of other labs have extended the so-called systems vaccinology approach to understanding immune responses to diverse vaccines administered to diverse human populations, including those at the extremes of age. These are some of the publications listed in the bottom left that we have published over the last several years, but really this approach is now quite widely used in the field to understanding and analyzing immune responses to vaccines. And some of the vaccines that we've focused on are shown here. I mentioned the yellow fever vaccine, but we've also been focusing on the seasonal influenza, avian influenza, malaria vaccines, and in the elderly, we're looking at shingles vaccines, and in the very young, we're looking at childhood vaccines. Another innovation that's happened in the field is the integrative modeling of multi-omics datasets. In this study, we analyzed the immune response to shingles vaccine, the Zostavax live attenuated shingles vaccine, and we analyzed the transcriptional profile, the metabolomic profile, plasma cytokines, and high-dimensional mass cytometry analysis. We were then able to integrate these orthogonal data sets to construct a multi-scale, multifactorial response network that revealed critical insights about the mechanisms by which this vaccine was inducing antibody responses. So in this talk, I'd like to show you one example of how systems biology can be useful in providing deep mechanistic insights about the human immune system. And I'd like to focus on what we call nature's endogenous vaccine adjuvants, and I'm talking about the microbiome here. And recent estimates are that if you calculate the number of genes in the humanity's microbiome, in other words, in the microbiome of every single living person on the planet, if you count the number of genes you have, these outnumber the number of stars in our universe. So our journey on this project began about 7 years ago when we analyzed the immune responses to the seasonal influenza vaccine using systems biological approaches. We defined a gene signature induced by vaccination early after vaccination, 3 days or 7 days, that correlated with the subsequent antibody response. And in particular, we observed that the expression of TLR5 or toll-like receptor 5 was highly correlated with the ensuing hemagglutinin titers induced a month later. Of course, you recognize toll-like receptor 5 is a receptor that senses bacterial flagellin. It was a bit of a puzzle as to why a vaccine against influenza should be inducing such a gene, and more importantly, why this gene was so strongly correlated with the ensuing antibody response. Jason O in my lab decided to investigate this puzzle, and what Jason did was to analyze mice in which TLR5, the gene for TLR5 was deleted, so-called TLR5 knockout mice, and he immunized wild-type mice or TLR5 knockout mice with the seasonal flu shot, and he saw that, in fact, there was a very profound impairment in the flu-specific IgG antibody response. Interestingly, Jason also observed a similar reduction in mice administered antibiotics or in germ-free mice, arguing for a role for the microbiome in orchestrating the antibody response to the flu shot. Of course, this was an experiment that was done in mice, and the question was, does this apply to humans? And why should one be concerned about this? Well, for many reasons. Firstly, as immunologists, we are curious about the mechanisms by which the human immune system functions and how it's affected by the microbiome. Secondly, from a practical perspective, of course, vaccines are administered widely to people in different parts of the world, and it's now becoming very clear that the microbiome composition of different populations in different parts of the world vary very widely, as shown here. Now, and yet, we all get the same vaccine. There are no personalized vaccines that are given to different segments of human populations. So the question was, do the differences in the microbiome in any way impinge on the immune response induced by these vaccines? To address this question, we performed a human clinical trial. When I say we, this was really the brainchild of a wonderful infectious disease physician by the name of Nadine Rupail. She is at Emory University, and together with her, we designed the study in which healthy human adults were administered a cocktail of 3 different antibiotics, neomycin, vancomycin, metronidazole for 5 days. On day 0, that is 1 day prior to the last day of antibiotics treatment, they were immunized with the seasonal flu shot. And then we analyzed the response for the following year. So these are stool samples were collected all the way up to 6 months, blood samples were collected all the way up to a year. And we did an extensive analysis of the change in the microbiome, but also the change in the innate and adaptive responses using various different assays shown here. The results of the study was published about 4 months ago, and I'd like to summarize the key findings from the study. The first question we asked was, what is the impact of this antibiotics administration on the microbiome? So we performed 16S ribosomal RNA sequencing to quantify the total bacterial load per gram of stool. And as you can see in red, these are the subjects who received antibiotics. There's a very profound drop in the total quantity of bacteria per gram of stool. And there's about a 3 to 4 log drop. It's about a 10,000-fold drop in the quantity of bacteria. What's very impressive is the rapidity with which this rebounds. Within a week or so, things are back to baseline level. Next, we analyze bacterial products in the stool. In this case, we're measuring the concentration of bacterial flagellin, or LPS, and this was performed in Andrew Gerwitz's lab. And you can see, similar to the total bacterial load, there's a very profound drop in the concentration of flagellin and endotoxin, but again, it's very transient and short-lived. Next, we analyze the impact of antibiotics on bacterial diversity. What's very obvious is that there's a remarkable constriction of bacterial diversity within a few days of antibiotics treatment, but unlike the rapid recovery in bacterial load that we saw in the previous slide, in fact, there's a much more gradual recovery, so that even at day 90 or day 180, the diversity that was observed at baseline has not yet been reached. So this tells us that antibiotics treatment has a very profound effect on the bacterial load as well as the bacterial diversity. However, whilst there's a very rapid recovery in terms of the bacterial load, there's a much more gradual recovery in terms of the bacterial diversity. So the key question is, what is the impact of this profound dysbiosis on the immune response? When we analyze the microneutralization titers induced by influenza vaccination, we observed there was absolutely no effect, despite such a profound effect, on the gut microbiota. Here we're looking at the microneutralization titers against the 3 different strains that are contained in the flu shot, the A California, A Texas, and B Massachusetts strain. This was measured in the laboratory of Hannah Golding and Suranda Khurana at the FDA, and the results were very, very clear. No impact at all on the immune response. Furthermore, we did a variety of different analyses, some of which are shown here. We fact sorted single plasmablasts and measured the somatic mutation in the immunoglobulin V genes with Patrick Wilson in Chicago. We analyzed the T follicular cell responses using flow cytometry and observed no differences. So this initial study was performed in 2015 in subjects who had relatively high baseline titers against the flu vaccine. Subsequently, in 2016, we performed a study in which we selected subjects who had very low baseline titers. We screened many subjects and we selected those who had very low baseline titers, and we excluded from the 2016 study any subject who would have received the flu shot in the 3 years preceding their enrollment into the study, or who would have had influenza infection in the 3 years prior. So what we were trying to do is to select for people in whom immune memory against influenza was minimal or relatively low compared to the average person. When we analyzed the antibody response in the 2016 trial, that is, in the trial in which we had selected for subjects with very low baseline titers and selected for subjects who had not received the flu shot or had been infected with flu in the 3 years preceding their enrollment, There we saw a profound impact on the antibody response. When Galit Alter's lab measured the H1N1-specific IgG1 binding antibody titers, there was a very profound drop in the antibiotics-treated people compared to controls. This was confirmed in Hannah Golding's lab by Surendra Khurana, and also in Hannah's lab, when they measured the microneutralization titers, there was a very substantial effect in the antibiotic-treated people. Furthermore, we also looked at the IgA, H1N1-specific binding antibodies, and consistent with what we saw with the IgG1, there was a major impact on the IgA, H1N1-specific antibodies also. So clearly, antibiotics treatment had impaired the antibody response against influenza vaccination in subjects with low baseline titers. What about the innate immune responses? We analyzed this using flow cytometry of analysis of innate cell populations in the blood, or using transcriptional profiling. And what was immediately obvious was that transcriptional modules involved in inflammation. So in other words, these modules contain many genes that orchestrate the innate immune response and the inflammatory response, were highly enriched in subjects who had antibiotics treatment. Why this was of great interest to us was that in a previous study that we published about 3 years ago, in which we had analyzed the transcriptional changes to influenza vaccination in healthy young adults versus elderly, we had seen that these very same modules that we were observing in the current study were highly enriched in the elderly subjects in the previous study. In other words, antibiotics treatment of healthy young adults had altered their transcriptional profile to vaccination to make it look like the transcriptional profile of an elderly person. In addition, we analyzed changes in the plasma metabolome using mass spectrometry. And in this principal component analysis, each line represents the metabolic trajectory of an individual, and each circle represents a time point. So, for example, you can see take any blue line and you can follow the line through its day 0, day 1, day 3, day 7. Interestingly, the blue lines and the red lines segregate very clearly. The red lines represent people who took antibiotics, and the blue lines are people who did not take antibiotics. In other words, antibiotics treatment has dramatically altered the metabolic profile in the plasma. In order to understand the metabolic pathways that might be perturbed by antibiotics, we use MummyChog, which is an analytical program developed by Shuzhao Li when he was in my lab as a postdoc. Basically, MummyChog allows you to go from untargeted metabolomics data to a pathway. And what was very clear was that there were major biochemical pathways that were perturbed by antibiotics, but at the top of the list was bile acid biosynthesis. To verify whether bile acids were in fact changed by antibiotics, we performed chromatography analysis, and we could see that in fact there was a major change in secondary bile acids, shown here. And this is one example of the extent to which secondary bile acids are changed. We're looking at the lithocholic acid here, and you can see there's about 1,000-fold drop in the concentration of lithocholic secondary bile acid in the plasma. And this is quite long lasting, and then at 3 weeks or so, things are back to baseline. So major change in secondary bile acids. Now, when we analyze the change in secondary bile acids shown here on the y-axis versus the total bacterial load shown on the x-axis, or concentration of flagellin or LPS, you see that there's a very profound and highly significant correlation. So in other words, the more bacteria you have in the stool, the higher the concentration of secondary bile acids. Now, what is secondary bile? Well, bile acid biosynthesis begins in the liver through metabolization of cholesterol, which gets converted into primary bile, which gets transported through the gallbladder to the intestine, where it gets metabolized into secondary bile by the microbiota. Secondary bile can bind to 3 known receptors, one of which is shown here, FXR or farnesyl X receptor, and mediate a range of biological activities. We analyzed the expression of the genes encoding the secondary bile acid receptors using the ImGen database, and we were very surprised to see that myeloid cells in human blood, including dendritic cells, monocytes, and other myeloid cell subsets, seem to express the highest levels of VDR and FXR. And so this raised the question of whether secondary bile could have an immune modulatory role by signaling through these secondary bile acid receptors. And to address this point, we analyzed the expression of genes involved in inflammasome. So the question is, how does this happen? How does antibiotics treatment result in enhanced inflammation and diminished antibody responses? Conceptually, you can imagine two distinct models. One is that you could argue that there's systemic access of PAMPs, bacterial products, caused by increased permeability of the gut epithelium. The second, you could argue there's an indirect effect of the microbiome on the immune cells in the periphery. So, to address the first question of systemic access, we measured the concentration of flagellin and endotoxin in the plasma and observed, in fact, there was no difference in controls versus antibiotics-treated people. This argues against a role for systemic access of these PAMPs. To address the second question of whether there might be an indirect effect, we performed an integrative modeling of multi-omics data sets, much like what I had mentioned to you before. And so we were able to integrate transcriptional profiling data with the metabolomics data, with plasma cytokines data, and the cell population. The results of our integrative analysis is shown in this slide. And in this network, what you can see is that the antibody titers shown on the top right and the change in inflammation shown on the left seem very distinct. They have very different connectivities. You see, the antibody titers are connected with metabolic clusters shown on the bottom right, largely, and some bacteria, whereas the inflammation modules are connected with some metabolic clusters, but also with secondary bile. In other words, there's a very tight inverse correlation between lithocholic acid, which is one of the secondary biles, but I could show you any number of secondary biles, and inflammation. But really, there's very little or no association between secondary bile and the antibody titers. Rather, what you see that is associated with antibody titers are metabolic clusters involved, enriched in fatty acid metabolism. This suggests that fatty acid metabolites might be controlling the antibody response to vaccination. And fatty acid metabolites might be the intermediary between the gut microbiota and antibody responses to vaccination. This is a hypothesis that we are thinking about testing in mouse models. So, to summarize, I think what we've learned from this study is that, in fact, the gut microbiota does have a profound effect on the immune system in humans, that as far as the adaptive immune response is concerned, in subjects with low baseline titers, that is, relatively weak immunological memory, the microbiota does act as an endogenous adjuvant. In other words, it promotes vaccine responses, antibody responses. In addition, we see that the microbiota, through the production of secondary bile acids, can control excessive inflammation. And so antibiotics treatment results in depletion of the gut microbiota, results in diminution of secondary bile, which ultimately results in an increase in inflammatory response. So this is what I've just summarized. The question we now have to ask is, what is the impact of the microbiome on immune responses to primary vaccination in adults or neonates and infants? As you can see, vaccines represent incredibly powerful probes on the human immune system. You can perturb the human immune system with vaccination, apply the tools of systems biology to analyze the signatures induced by vaccination, perform bioinformatics analysis, and generate interesting new hypotheses about the mechanisms by which these immune responses are generated and maintained. We can test these hypotheses using animal models, and eventually the understanding and insights that we gain from these mechanistic studies should and could inform future vaccine design. The particular example I've shown you in this presentation is our work that began by analyzing the human immune response to the seasonal influenza vaccine in which we discovered that the expression of toll-like receptor 5 was induced and highly correlated with the subsequent hemagglutinin titers. We then tested the causal effect of toll-like receptor 5 and the antibody response to flu vaccine in the animal model, in mice, and we discovered that in fact TLR5 did play a critical role in the immune response to influenza vaccine, and in fact this was because of its sensing of the gut microbiota. We then performed a subsequent study in humans to analyze the impact of the gut microbiota on immune responses to vaccination, and in that study we discovered that bile acid metabolism, in other words secondary bile acids, were critically involved in regulating the innate response. This has become a project in the lab in which we're investigating the impact of secondary bile on immune responses to vaccination, and perhaps in the not too distant future, in the next couple of years or so, we can think of us and perform an experiment in humans to evaluate the impact of this bile acid pathway on immune response to vaccination. So let me conclude by thanking the people who did the work in my lab. This was led by Thomas Hagen and Mario Cortez, two outstanding postdoctoral fellows. As I mentioned, this was a close collaboration with Nadine Rupail's lab at Emory, as well as with Galit Alter's lab, Hannah Golding's lab, Patrick Wilson's lab, and Andy Gowitz's lab, with generous funding from the Human Immune Profiling Consortium at HIPSI. Thank you for your attention.
You are muted. You are muted.
Thank you very much, Dr. Pulendran. We will now go on to the two lectures selected from the abstracts. I'm going to introduce both of them, then we'll have questions on all the talks at the end. So the first is Alessandra Borgognone. Her title is Baseline Gut-Associated Microbial Signatures May Help Predict HIV-1 Viral Control After Treatment Interruption. And the second one is Shamsudheen Moidunny. His title is Gut Microbial Regulation of Anxiety Disorder in a Murine Model of HIV-1 and Opioid Abuse. So, Alessandra.
Good afternoon, everyone. My name is Alessandra Borgognone. I'm a postdoc at the Microbial Genomics of IrsiCaixa, and I'm going to present the results of an exploratory study focused on the impact of the gut microbiome in HIV vaccine response. As you may know, the gut microbiome is affected by several factors, and in turn, it is constantly modulating the host immune system, thus representing a crucial component that might affect how individuals respond to vaccination. However, investigation on the relationship between the gut microbiome and vaccine efficacy are limited so far. In the context of HIV-1 vaccine, this sub-study derived from the VCN-02 clinical trials designed by Christian Brander and Bea Mote, where a kick and kill strategy was assessed by combining MVA vaccine administration before and after 3 doses of romidepsin, here used to stimulate the viral reservoir, and followed by a monitored treatment interaction for 32 weeks, where the treatment was stopped to monitor the viral replication and also measure if this immune strategy actually worked or not. The trial ended up with 3 out of 13 participants which achieved HIV control, meaning that they were able to control viral replication without treatment. And in this sub-study, we want to investigate if these 3 individuals able to control viral replication had different composition in the microbiome at the baseline. This is a methodology, the review of the methodology used in this study, where longitudinal stool samples were analyzed using shotgun metagenomics for the gut microbiome characterization, whereas PBMC transcriptomic profiles, inflammation-related proteins, and fecal proteomics were examined at the baseline. We found that individuals able to control viral replication had very high levels of bacteroidales when compared to non-controllers, whereas clostridioides were significantly reduced. This was at the baseline. Even before vaccination and during vaccination, more or less, this association remained. At the family, genus, and species levels, we can see that non-controllers were enriched in bacterial groups from Clostridiales, the traditional associated with lower inflammation, such as Subdoligranulum and Roseburia, which is a butyrate producer, whereas HIV controllers were enriched, for example, in Revotella, suggesting that individuals not able to control viral replication had less inflammatory, presented less inflammatory bacteria in their gut microbiome composition. If we look at the longitudinal Bacteroidales and Clostridiales abundances and also their ratio, we can see that the Bacteroidales Clostridiales ratio remained increased in individuals able to control the viral replication, that the gut microbiome composition seems to remain relatively stable over the trial. So this we will have a pretty clear signal associated to the gut microbiome composition, and we wanted to further analyze the baseline to identify additional signature that might support this observation. The baseline PBMC transcriptomic analysis showed that genes upregulated in controllers were enriched in functions associated with immune activation and inflammatory response. In addition, inflammation-related protein biomarkers were also overrepresented in this group. So these results seems to indicate higher inflammation and immune activation in controllers in which anti-inflammatory bacteria were less represented. The preliminary analysis of baseline fecal proteomic profiling showed that differences between these two groups were mainly driven by Clostridiales, and we also observed that despite being less represented in controllers, Clostridiales were more activated in this group, as shown by increased protein levels involved in different cellular metabolic pathways. So we have to further elucidate mechanisms explaining these results. But from results obtained until now, we can conclude that a higher baseline Bacteroidales Clostridiales relation may help predict HIV viral replication after treatment interruption, and that pre-vaccine immune activation and inflammation potentially linked to lower anti-inflammatory bacteria may contribute to enhanced vaccine response. So we have a clear signal, but in a very small study. They actually need to be validated and confirmed in independent and placebo-controlled study. In particular, we need to repeat this study in larger vaccine, in this analysis in larger vaccine studies where we have enough individuals able to achieve viral control during treatment interruption. So this is ongoing in our unit, and elsewhere. In the meantime, we are finalizing the metabolomics and fecal proteomics analysis in collaboration with Adam Burger and Rafik Sekali. So we will certainly require external validation in other clinical trial, but if these observations are confirmed, we could potentially predict who could control HIV replication beforehand, and maybe select more precisely which patients may better benefit of these treatments, and in the future try to develop interventions that might convert non-responders to responders by manipulating the baseline gut microbiome. I want to acknowledge all the people involved in this study, and thank you all for your attention.
Hi, everyone. I want to start by thanking the committee for selecting my abstract for an oral presentation. I also want to thank Federica Ressa for coordinating this meeting. Today, I'll be talking about one of my ongoing projects at the Department of Surgery in University of Miami. The title of my presentation is Gut Microbial Regulation of Anxiety-like Disorder in a Murine Model of HIV-1 and Opioid Abuse. Anxiety disorders have been reported to be as high as 50% in HIV-infected individuals. Anxiety disorders often coexist with major depression in HIV patients. In fact, those who suffer from anxiety are 50% more likely to suffer from a major depressive episode. Moreover, the prevalence of anxiety and depression may be greater in those who are substance abusers. A number of recent studies have indicated an important role for the gut microbiota in mental health as well as in the development of cognitive and psychiatric disorders, including dementia, depression, and anxiety, through the bidirectional microbiota gut-brain axis. The gut microbiome can regulate brain function by either producing neurotransmitters, including GABA, which has potent anxiolytic effects, by regulating immune function, by modulating vagus nerve transmission, or by producing various other metabolites such as short-chain fatty acids. In a recent study by Bravo et al., Lactobacillus rhamnosus treatment was shown to alleviate stress-induced anxiety behavior in mice. And these effects of Lactobacillus rhamnosus was dependent on vagal afferent transmission. We recently observed a significant change in the gut bacterial communities in mice following 24 hours of morphine treatment, and this was associated with a significant depletion of Lactobacillus bacteria. As you can see, the big blue bars in the control groups, which represents Lactobacillus, are almost completely wiped out in the morphine group. We also observed that the morphine-induced gut bacterial dysbiosis was more severe in the context of HIV infection. In BLT humanized mice, HIV and morphine independently and additively induce gut dysbiosis, as shown in the graph on the right-hand side. In addition, we observed a distinct bacterial clustering in HIV plus morphine group compared to HIV or morphine alone. Similar findings were also observed both in opioid-abusing HIV patients and in SIV-infected monkeys chronically treated with morphine. In BLT mice, the morphine plus HIV-induced gut dysbiosis was also associated with gut epithelial barrier disruption, translocation of gut bacteria into the peripheral organs like liver, and induction of systemic inflammation. Since recent studies showed that anxiety and depression in HIV patients is associated with elevated plasma immune activation, we hypothesized that HIV and opioid-induced gut epithelial barrier disruption can contribute to systemic inflammation and development of anxiety and depression, especially in opiate-abusing HIV patients. To test this hypothesis, we used the HIV TG26 mouse model, which expresses 7 out of 9 proviral genes. This mouse model has been suggested to partially mimic conditions of the virally suppressed HIV patients on ART, since it expresses HIV viral proteins in the absence of viral replication. In addition, recently we established TG26 mouse as a suitable model to study HIV-associated cognitive impairments. In this study, we identified sex-dependent impairments in hippocampal-dependent contextual fear memory only in male TG26 mice but not in females. The memory impairment in this mouse model was associated with an increase in glycogen synthase kinase 3, a serine threonine kinase enzyme. We also identified impairments in hippocampal synaptic transmission and molecular deficits including reduced glutamate receptor activation, reduced BDNF levels, reduced synapsin levels in this mouse model, which were all dependent on GSK3. We therefore next sought to use this mouse model to study anxiety behaviors using the open field test. This test makes use of the innate aversive behavior of rodents to a novel environment. Typically, an anxious mouse would spend most of the time along the sides of the wall or the outer zone, indicated in blue, whereas a less anxious mouse would come out to explore open area or the inner zone, which is shown in green. The degree of anxiety in animals can be measured with multiple parameters, including the time spent in the inner zone or the frequency of visits to the inner zone. For this study, we used a total of 4 groups of mice, littermate wild-type and TG26, which were treated either with saline or morphine. On the right-hand side is a representative heat map of 1 mouse per genotype following saline or morphine treatment. As you can see, morphine-treated TG26 mouse, which is the bottom right image, showed very little time in the inner zone compared to all other groups. This is indicative of anxiety behavior in TG26 mouse following morphine treatment. Upon quantifying anxiety behaviors in a group of 8 to 10 mice per group, that is the time spent in inner zone and the frequency of visits made to the inner zone, we observed that the TG26 mice displayed anxiety-like behavior, which was further potentiated by morphine treatment. Additionally, we observed that morphine treatment in TG26 mice induced freezing behavior and reduced locomotor activity. These effects were not observed in wild-type mice after morphine treatment. These findings suggest that the TG26 mouse is a suitable model to study HIV and opioid-induced anxiety behavior. Ongoing studies will use this mouse model to develop symbiotic approaches to alleviate anxiety-like behavior. In addition, we are currently doing a multi-omic analysis to identify novel pathways associated with anxiety disorders in this mouse model. Finally, we will test the effects of anxiolytic effects of engineered bacteria in this mouse model. With that, I come to the most important slide of this presentation. I want to thank Dr. Sabita Roy for all the support and guidance. All my wonderful colleagues at Roy Lab, both present and past members. My collaborators at University of Minnesota, University of Miami, especially Eleanor, Colleen, Titus, and Michael. Funding agencies NIDA and Miami CFAR. Thank you all for listening, and I will take any questions.
Okay, thank you all. Those were really fantastic presentations, and there are a number of questions here. I can just get started with the first one for Shamsudheen. Have you looked at the gut tissue section? So any kind of changes in enteric nervous system in these mice?
So not particularly in these mice, but we've looked at in wild-type mice. This is like a ongoing study, and what we've seen is an increased infiltration of immune cells following morphine treatment. So what we expect is in an HIV mice, we would see more of it. So yeah.
Okay, thanks. So the question is for Alessandra, and this is from Alan Landay. What happens in therapy interruption without vaccines? And the question is, does microbiome predict post-treatment control?
Thank you for the question. So we don't have evidence for post-treatment control without vaccine, for that a certain gut microbiome may predict post-treatment control. But for sure in future validation studies, we may consider include additional arms to validate this possibility. Yeah.
Wonderful. It was a great presentation too.
Thank
There are several questions for Bali, but I would like to just start with one. So Bali, the changes that you're talking about, and this very provocative, nice hypothesis that fatty acid metabolism may be playing a role and bile acid metabolism. Many of these changes are seen in HIV infections, some of the other chronic viral infections. So it is great that antibiotic treatments induce some of these and you can rapidly generate those changes, but how do you see that impacting vaccine response? Are there some common fundamental mechanisms here? You are muted. You are muted.
Sorry. Okay. Can you hear me now?
Yes.
Okay, very good. Yes, it is a good question. We see all these changes happening. At a mechanistic level, we're very interested to see how these changes in bile acid, secondary bile acid, for example, how this impacts vaccine responses. So we are beginning experiments in mice where you could use knockout mice in which specific bile acid receptors are knocked out, TGF5 knockout, for example, or you could supplement mice with different kinds of bile acids. As you know, there are so many secondary bile acids, and really explore what they do in terms of a vaccine response. And I think Jay Bazovsky had a question whether these affect the antibody response, and those are experiments that we're doing. If there are any young people looking for a postdoctoral position to come and work on these questions, we would love to invite them. So anyway, so that's what we're doing, and but we don't have any real mechanistic data as yet. So.
Okay. There was another question for you. That is, what drew you to focus on subjects with low pre-existing antibody titers?
It was complete. It was complete failure and a lot of heartache after the 2015 experiment. We despite such a huge perturbation in the microbiome, there simply wasn't an effect on the antibody response. And so, of course, then the obvious question was, well, what happens if you redo this experiment with people with relatively low memory? And that's why we selected for these people with low pre-existing antibody titers. I mean, we had this at the back of our minds going into this experiment in the first place, because we thought, yes, the microbiome probably has an effect, but a strong enough vaccine with a strong adjuvant, or if you have a lot of immune memory, it's probably able to withstand even a very drastic effect on the microbiome. But that's what led this led the second experiment. And in fact, right now we're doing an experiment with the rabies vaccine in people who are completely naive to the rabies vaccine. And that's a really good primary response. That the data is still emerging, but it really looks like there's a really good effect in terms of the antibody titers there.
So Alan Landay has a question for you. There are significant changes in microbiome with age. So have you sorted these out in your
Yes, that's a good question, Alan. I mean, we're very interested in the extremes of age. I mentioned neonates and infants mainly because a lot of the childhood vaccines are given during the first year of life, and the microbiome is very nascent and it's evolving in the first year of life. So, and unfortunately, many children in the developing world, I mean, there's rampant use of antibiotics, as you know. In many developing countries, unfortunately, physicians prescribe antibiotics quite a lot. And so what impact would that have on the responsiveness to childhood vaccines? This is a study that we're doing with our collaborators in the Gambia and also in India, South India, in Vellore, to explore this. But at the other extreme of age, in aging, as you know, this whole phenomenon that's been called inflammaging, that's been associated with aging in many in people with the Western diet and these sort of inflammatory diseases. And so the question is, how does that impact vaccine responses? And given our data that the secondary bile acids seem to be controlling inflammation, does this secondary bile inflammation axis come into play during inflammation, in the elderly? Those are issues we're interested in as well.
One question from Laurel. Do particular antibiotics such as vancomycin may have greater impact on bacteria? This is for converting primary to secondary bile acids.
Yes. We don't know, Laurel. We haven't done the experiment, and I'm not sure. I mean, we thought initially we would give the cocktail just so that we could see, we wanted to stack the odds in our favor of seeing some effect, and we figured that the cocktail would have a profound effect. But you're absolutely right. I mean, maybe different classes may have different effects.
Right. Some few questions for Roger, and I would like to know this too. So why do you think that changes in magnitude mostly occurred in female mice?
That's really unknown. So what the first thing we are doing is, so there could be some innate differences, and we really want to explore that with more detail. I have to warn you that it's a limited set of, it's only 5 patients, 5 mice per gender, and then we are repeating everything. But but there seems to be some innate differences possibly in, and we need to understand that a little bit better. Yeah.
Okay. This question from Kathy Lazapan. From their lab, they reported that Holdimanella, you may already know that question and answer, Bioformus, they showed that it induces CCR5 expression on T cells. And so she's wondering whether you've shown erysipelotricalis, whether it falls in the same group, which were lower in controllers compared to non-controllers, whether it's a similar mechanism or.
So we see an increase in Bacterium in non-controllers, and we don't have Bioformus, so we have other. We see increases in when we've done 2 species we see it mostly in the Bacterium rectalia and Cireum, but it's certainly a possibility, and I will go back to the data and look a little bit deeper, because we have to really. I think it's a very good proposal, and we really have to go a little bit deeper. The limitation with the study, I mean, we are doing a lot of fancy things in very few patients, so it's only 3 patients. So we, at the end of the day, we need to confirm this in a much larger data set. But the difference is so big, and also it makes sense. I mean, everything else. You see in the transcriptomics, as Alessandra was explaining, a signature in innate immunity and neutrophil, also a neutrophil immunity, base immunity. So I think it's very interesting to propose. But we'll look into Bacterium before me.
Okay, I think I'm supposed to now turn over to the next group to summarize, and don't see we haven't missed any questions here, but outstanding presentations both from established investigators and our junior investigators. That was great. Thank you all.
Thank you.
Great. Thanks very much, Satya. Let me now hand it over to our final speaker, Grace Aldrovandi, for closing remarks.
Hi, I'd like to thank on behalf of the co-chairs my tremendous gratitude to all our speakers and abstract presenters for sharing absolutely phenomenal data that I know has stimulated me and everybody else to move forward in our studies to try to understand how these microbial communities intersect with HIV and human health. I want to give a shout out to Kunchi and Frederica for organizing this virtual meeting. I think this is as good as it gets in a pandemic, but I would like to move forward. I'd also like to acknowledge the organizing committee that helped put together this program. Kunchi, are you putting up the slides? I can't see the slide with our organizing committee that brought together this outstanding panel of speakers. And Roger, mute. I think we were violating HIPAA. So we do have pictures of the a snapshot of the organizing committee. Is that being projected? I don't see it on my slide set. Okay. Well, it should come up at some point. And we do have a group photo of everybody that was taken virtually, kind of for Americans of a certain age. This is kind of the Brady Bunch on steroids. I want to thank our endorsers, our non-commercial sponsors. Oh, it looks like we're catching up. And our industry sponsors who help subsidize this. And I'd like to point out that next year, hopefully, we will be meeting in person and at the NIH. Tentatively, the date is, I can't see it, but I believe it's October 20th and 21st. So we want to not have virtual cocktails, and as an Italian, I have to hug everybody. So my final words is thank you, stay safe, and stay sane.